whether serious or very, very ridiculous, is really important to help us out as scientists. So, kind of when I was thinking about doing this, I thought this was a great idea. And then about 2 weeks before this happened, I thought, what was I thinking? Because I have no idea what I'm doing. Netflix, you can binge watch all of, all the comedies they have. It's not going to help you out very much. One thing I thought about was that maybe I'd have a 60 to 80% bomb rate. And I was kind of upset about that, but then I talked to my lab and they were like, wow, Teddy, that means you have a 20 to 30% success rate. That's like incredible in academia. How could you possibly be upset about that? And that actually made me feel a little bit better, and also really sad. But so, I think tonight this is going to be a really great opportunity. Instead of being very serious, trying to impress everybody, show you some nice pretty graphs, I can drink my beer, make fun of myself, and really just impress nobody. So, before we start, I have 3 main rules. The first rule, please be kind. This is my first time. Like I said, I've only binge-watched comedy. I have not been up to an open mic at all. And so, if I wanted to be ridiculed, if I wanted my confidence stripped, if I wanted basically a high expectation of failure, I would have stayed in my lab and worked my grant. So, rule number 2, if you do feel like you want to, be a heckler, that's fine. I have a nice corner over there. It's called reviewer number 3. Please don't be a dick. And for all those of you who don't really understand what I'm talking about, in academia we have the peer review process. It means that you have 2 reviewers that are nice, they're friendly, they give you constructive feedback, and then you have a dick. And quite honestly, they're like Joker from Batman. They just want to watch the world burn. And so they're kind of like a mythical sphinx. You're on your path to your adventure, and they stand in your way. They give you weird cryptic clues that you have no idea how to figure it out. And if you don't solve any of them, they eat your heart out. So they're terrible. And then rule number 3, we're at a brewery. We have beer. This is my confidence beer. Hopefully it stays with me and doesn't run out before I finish, because we'll see what happens with that. You all have your own beer. And hopefully if our beers stay filled, this will be a good time. So, kind of starting off with this, why did I become a scientist? I'd say the first time I really thought that I wanted to go into science was in elementary school. And I'm a microbiologist, so the first thing I thought about was the big epidemic we had, especially in the cafeteria, the cooties virus. And I was kind of a weird kid, because you have all the boys that want to stay away from the girls and they're afraid of getting their cooties. I'm the guy it's not a Rugrats notebook, I'm not going to spend $10 on that for a prop. I'm the guy with my Rugrats notebook, kind of walking up, looking at the girls, trying to figure out what's going on, because quite honestly, there are no symptoms of cooties. I don't know how it's spread, how it's transmitted. All the boys are telling me to stay away from the girls. I don't understand that. I look through my notebook. There's no evidence. There's no peer review process. And quite frankly, the biggest thing that I have no idea how it comes about is the cootie shot. You can solve all your problems with circle, circle, dot, dot. And that doesn't make any sense at all. I mean, let's be honest with it. If that was how vaccines worked, this would be a better place. And I was also thinking, for boys who really believe in cooties, why don't they just tattoo circle, circle, dot, dot on their arm and call it a day? It doesn't make any sense. But the one that really doesn't make sense is circle, circle, square, square. Now you have it everywhere. Because that is the most terrifying zombie apocalypse I have ever seen. Because you're talking about the spread of a pathogen through just very terrible 10-year-olds. People say you have an estimated 48-hour window to spread across the world. This thing is going across in 12. Those kids are just going to be out of spite telling everybody that they have it. And so, kind of looking back on all of this, it's really interesting because for being a kid, having absolutely no evidence about cooties, we don't have a peer review process, we don't publish, you've never seen a link between the cooties vaccine and autism found in PBS kids. It's pretty bad. So, that's kind of how I started at it, and then I'd say the next thing was really finding a science role model. And a lot of people, they think that their first role model, I've heard a lot of people say, is Bill Nye. And that's great. I like Bill Nye. But all of his experiments worked. He's switching from lasers to tornadoes to all these different experiments, and that's not real. My role model, it's Q from the Bond series. That guy is incredible. He works for 10 years coming up with this cool prototype. He does all these amazing research. He does all this testing in the background. You see gunshots going off and bulletproof vests. And then, of course, Bond blows the shit up. I mean, that's dedication. And he's also the one guy in all of the series who doesn't put up with his shit. Because you have, the henchmen, a karate chop, and they're down. Girls are in bed the next morning, he's off. And then you see Q, where it's just Bond, I've worked on this experiment for 10 years. This car is perfect. You can remotely activate it. You can do all these things. Just please bring it back in one piece. And then you have Bond very smoothly, I'll do the best that I can. And he's just... Because that's what science is about. It's really spending all the years, and then you have a Bond that just throws it over the river, throws it up, and you're like, all right, I'm back to square one. But he's a hero, and don't forget that. And so, kind of now going forward, I've wanted to be a scientist, and I decided that I think I really just wanted to be a microbiologist. You heard my thing about cooties. I think I'm really interested in the microbial world, and I think that's what really got me into a lab about bugs. I work with wasps. It's not medically based, it's not humans. And there's some problems with that. It's very hard to tell my family of my self-worth when I say I switch bugs all day. And, it's kind of hard because you're in a crowded area, someone falls to the ground, no one's yelling out, is there a microbiologist around? And you're running over there, you pull out your microscope, you put some blood on the slide, it's like CSI, you're looking at it and you're like, all right, everybody, there's an Ebola outbreak. It doesn't happen like that. You don't really get the public support. You're not curing cancer, you're not producing patents that you can sell off to pharma for billions of dollars. But bugs have their benefits. For starters, a common thing in research is that you have to have protocols about how you humanely handle your animals, and it makes a lot of sense, but it's kind of nice that, under my protocol, it's just squish, It's really easy. And so another benefit is that you really get the benefit of the doubt and flexibility to really just name genes whatever you want. And I have a list of some really good ones that I really enjoyed. Some of them start off a little, mild. You have rutabaga, radish, turnip, cabbage. They're all about learning and memory. It makes sense. Those vegetables have been known to help with those kinds of things. Then you get a little bit more morbid. You have Tin Man. It's a Drosophila mutation that means that you don't develop a heart. It's a little sad. Then you have another one. You have the Lush gene. It means it's a mutation that makes you Drosophila unusually attracted to ethanol, which, some people might call that alcoholism. And then finally, for the Monty Python fans, there's Indy, the I'm not dead yet gene. Because if it's mutated, you actually live twice as long, though, it's being discussed right now. And then you get to just the crazy mad scientist genes that you're like, did this come out of human centipede? Because there's one called Ken and Barbie. And this is a Drosophila mutation where they have no outside genitalia. And my first thought is that not only did you see this phenotype, which it happens, you throw some x-rays at the Drosophila, they create crazy mutations, but the fact that you went on to study this so long that you came up with a gene for it and then decided to name it after your kids' dolls. It's a little messed up. And then you get a little bit tricky. You have one called Cheap Date, and this one is a mutation in alcohol processing. So it means the Drosophila gets drunk quicker and it does not go away fast.
What's Drosophila?
Drosophila are fruit flies. You see them everywhere. They're very annoying and they mess up your fruit. You're welcome. And so going with these genes, it makes a lot of sense that they don't exist in humans. Because it's really hard to imagine a doctor walking into your patient's room, you sit down all serious, and then you have to consult them and say, "I am so sorry, Mr. Johnson. Your neurodegeneration is unfortunately due to a mutation in your Swiss cheese gene." It's very sad. If you'd like a description of it or a visual, here's your MRI. It's pretty bad. And so, I'd say there's kind of some benefits to sticking in the fly world, squish, fun gene names. I unfortunately was not creative enough. I spent 6 years on a project and I came up with the literal name of what the gene did. And I kind of regret it now because I had so many possibilities. But it happens. And so, just to conclude, I'm going to finish with something that I do a lot in academia, which is write poems. This one is an ode to grad school. When you come to grad school, you learn a whole lot. Your friends found jobs with good money, but alas, you did not. You studied science, and knowledge is your payment enough. This is why there are grad students whenever there's free stuff. In lab, you study day and night. It only takes 5 years. You get it through it with your friends, and many, many beers. Because working in the lab, you collect data page after page, and working at least 60 hours a week, you barely make minimum wage. And then you think, I got this, surely I know a lot. But through an exam with a committee, your confidence is shot. But eventually, you finish your first project, it worked, yippee. Screw a paper in a defense, can I please have my PhD? No, there's so much to do with talks, papers, and grants. No quality time with your partner, or even a text with eggplants. But things pick up steam. You begin to shed your doubt. People naively think you actually know what you're talking about. Then the magic happens. Your committee says you can go. You plan your final defense. There are so many ragers to throw. You did it. No more does your defense seem scary. You can get to dress in regalia. You're a wizard now, Harry. Some might say a professorship is where you should go. But since I'm here, right now, trying stand-up comedy, I'm thinking my answer is hell no. So thank you all very much.